Okay, everybody, it looks like we've got a good start time for 10 o'clock Pacific time and 1 p.m. East Coast time. Thank you for joining our webinar today. My name is Tom Driscoll. I am the Vice President of Marketing at Fluxion Biosciences, and I'll be reviewing today information about a new product called CytoFlux, an imaging cytometry system. So first, a little bit of background on Fluxion Biosciences. The company was founded in 2006, headquartered in South San Francisco in California. We have worldwide distributors throughout Europe, Asia Pacific, as well as partners in the United States and Canada. The company is a winner of 5 NIH grants and a 2-time R&D 100 award winner, first for the BioFlux System in 2008, and second for the IonFlux System in 2010. A common feature and theme and design throughout all of our products are microfluidic designs that simplify and enable complex live cell assays. We have 3 product families. The first one that I'll review on the bottom is BioFlux, which is used for live cell assays in microscopy applications. The next product is IsoFlux, which is used for isolating circulating tumor cells from whole blood. And I will review some of the CTC enumeration as an application later in the presentation. And the third product family is IonFlux, which is used for automated patch clamp applications, primarily in drug discovery. So our agenda today, I will give an overview of the workflow and the individual components and how we've leveraged our BioFlux product and the designs in that product to enable an imaging cytometry capability. Next, I'll review a bead dilution where we've diluted 5 microliter aliquots of green fluorescent beads to establish linearity of the system. And then I'll go into 2 specific and very different applications. The first one is cell cycle analysis of pubic cells that were plated in a BioFlux plate. And this is a good example of showing the analysis of live adherent cellular analysis, something very, very different than what a typical flow cytometry system could provide. And then the second application will be the CTC enumeration as an extension of our IsoFlux system and what we believe is a better way to perform CTC enumeration. So the CytoFlux system is an imaging cytometer that uses the BioFlux components that enables uncompromised image quality. And we do that because the system takes advantage of your existing inverted microscope. So the image quality that you get with your microscope, whether it's a Nikon or a Zeiss or an Olympus, it really doesn't matter. Whatever your microscope setup is, we can take advantage of that image quality. In addition to that, the system setup can enable this type of analysis with both adherent and suspension cells. We can also do this with minimized dead volumes, and I'll show the 5 microliter aliquot data, as well as the ability to recover your samples after you've gone through the image capture and the analysis. 4 very unique capabilities and very different than traditional flow cytometry. So the core technology of the system is centered around what we call our well plate microfluidics. What we've done is we have designed microfluidic channels into the convenience of a traditional well plate format. That format works on any inverted microscope and is suitable for both automated and manual fluid addition. The microfluidic format capability enables minimized dead volumes with reagent additions as low as 5 microliters. And the other major benefit to using the microfluidic channel is a very smooth, very controlled laminar flow. And ultimately, that system assembly provides us high content data within a flow cell to provide plate-based microfluidics with high resolution microscopy. As the diagram in the lower right-hand section indicates, cells can be pipetted into a single well, and we use pneumatic pressure to gently push the cells from one well to another. And those cells travel through a microfluidic channel which is then viewed through a microscope objective on the bottom. And I'll go through an application with a HUVEX that shows how those cells are adhered to the inside of that microchannel, and images can be viewed in high resolution. So an overview of the CytoFlux protocol and how the workflow works. With suspension cells, we use what we call a flow stop image protocol. So the software of the system controls the microfluidic regulators and pumps to provide variable speeds so we can go fast and slow. And we also provide the ability to have timed delays so that the cells in the microchannel can flow, be stopped, and then imaged, and then start to flow again to remove those cells from the field of view, and then take the images of the next cells. The software also provides control of the stage movements on the microscope, as well as all the optics and the camera so that we can capture the images in whatever resolution or speed we need. The third piece of that workflow is once the images are captured, the image processing to define the individual objects, detect the edges, and reduce all of the images to data. And then that data is formatted and handed off in a format so that it can be opened in a software package called FCS Express from our new development partners at Novo software, so that the image can now be analyzed and treated just like traditional flow cytometry data. So we'll show some examples of how that data can be used to visually verify your individual cells. It can also be used to go back and forth between your dot plots as well as your images, and also the ability to facilitate an automated final analysis and report. In the BioFlux plates that we can use for the suspension cell captures, the microchannels were designed to provide a nice, smooth, laminar flow. So in the process of image cytometry, what we want to be able to do is have a nice, smooth, controlled flow of the cells through the microchannel and the ability to stop those cells for imaging. The original BioFlux plates were designed to mimic in vivo environments in the vasculature. So just as you would expect a smooth flow through the vasculature, the plates are designed, as you can see on the right, to have a nice smooth flow in the velocity. The dimensions of the channel are approximately 370 microns wide by 75 microns tall. So depending on the cell type, most mammalian cell types do not have any problems with those channel dimensions. So depending on the individual application, some larger cell types will have problems in that particular dimension, but for the majority of mammalian cell applications, those channel dimensions are just fine. So our first application summary that we'll go through is the dilution of green fluorescent beads to establish linearity. So we ordered standard fluorescent beads, I believe, from Bang's laboratory, nothing special. We diluted these into a 8-point dilution to get 4 orders of different signal intensity. And ultimately what we see here with 5 microliter aliquots that were pipetted into the individual wells in the BioFlux plate. We sent those beads through a series of 40 stop flow steps, which ultimately captured tens of thousands of data points. And when the data was the images were automatically captured, the data was automatically reduced, formatted, and sent into FCS Express, where a template automatically populated the bar plots to establish the linearity data. Once the beads were pipetted into the plate, the process takes about 5 minutes or so to capture the images and then have them automatically sent over to FCS Express for the automated analysis and summary. So with the traditional CCD camera, in this case it was an off-the-shelf 1394 Q imaging camera, we established just about 4 orders of magnitude of linear signal. So the first biological application that I'll provide an overview for is the cell cycle analysis of the adherent HUVEX in the BioFlux plate. So we subjected the cells to two different drugs, an agonist and an antagonist. The agonist is a very well-known and documented fibronectin growth factor, pleiotropic growth factor that controls proliferation and overall differentiation, enhances it enhances the population doubling without altering the phenotype, and we'll have some good data to show that. So what we were primarily looking to do was to verify published data with both the agonist as well as the antagonist. And we chose the antagonist, a very interesting compound called bicalene. It's a flavonoid, which is in very many herbal medicines. And there are different publications that show some different results, as will typically come with most drugs. In high concentrations, some drugs can be toxic, and in low concentrations, they can have different effects. So the two different things that we were concerned about and interested in were the ability to inhibit cell proliferation, and specifically the inhibition of angiogenesis and the suppression of tube formation, in HUVECs in particular. So what we wanted to measure were the cell cycle arrest in the G1 and the S phase. So those were the two pieces we were looking for. With the agonist, enhance the population without changing the phenotypes, and in the antagonist, getting the measurement of the G1 and S cell cycle arrest. A quick overview of the protocol. For our reagents, we used a simple nuclear stain of Hooks. To look at the cytoskeleton and the structure, we had an Alexa 48 colloidant to look at the actin. For the reagents to fix and firm the cells inside the plate, we used simple PFA and Triton, and then simple buffer, ultimately to do some of the washouts in the microchannels. For the BioFlux plate, we established a monolayer by pipetting live cells into the outlet well, and we draw those live cells back into the viewing channel as indicated in the graphic in the right-hand side of the screen. So cells are pipetted in, and we literally use the pneumatic pressure to push the cells back into the viewing area. Once those cells are lined into the microchannel, we use a buffer, just simple PBS, to wash out any extra cells and incubate the cells overnight to establish the monolayer within the microchannel. Cells were then subjected to 8-point dilution series that were centered around documented and published EC50 and IC50 results. Cells were then subjected to shear flow of 2 dynes per square centimeter for 16 hours. The cells were stained and then imaged, which leads us to an initial data set. So this table of images are representative images for IC and EC50s from those individual dilutions that we showed. And these are, I think, great indicators of exactly what we expect to see, where we see biokaline as clearly a compromise of the actin and the cytoskeleton on the left-hand column. And on the right-hand side, we see normally healthy cells with a strong cytoskeleton. And in particular with the FGF, what we also see is an increase in the number of cells that are dividing, ultimately an extension and an increase in the number of cells going through cytokinesis, which is exactly what we would expect with an FGF. So transitioning those images into data, once the images were analyzed and the data was reduced and transferred over to FCS Express, the data was populated into a cell cycle graph. The y-axis values were normalized to the count of cells, and here we saw exactly what we expected from the images was an extension of the cells going through cytokinesis. So that we thought was a great verification of exactly the published data that we saw with the agonist. For the antagonist, what we wanted to do was to duplicate the publication results that are indicated in the lower part of the screen. In particular, what we expected to see with the biokaline on the HUVECs in the G1 and S phase arrest was a decrease in the S phase and an increase in the GO and G1 phases. So as you see with the, if you call those burgundy bars, we see a decrease in the S phase concentration or percentage. What we also see by looking at the blue bars, you see an increase in the GO and G1 phase. Exactly what we expected and a great duplication of the published results. And the last piece on this particular data set that I'd like to share is a 3 data set overlay which has a very unique characteristic that one could not get with traditional flow cytometry. So the individual data plots, or the dot plots, are color-coded for the treatments. Treatment 2 is the control. Those are the black dots. Treatment 1 is the FGF. Those are the red dots. And treatment 3 was the biokaline, which are the blue dots. So what we can clearly see, ultimately we had about just under 2,200 cells that were captured for this analysis. But what we can see here is a clear differentiation of subpopulations, even within a relatively small data set of about 2,200 cells. What we found interesting is the total nuclear size was significantly increased with the biokaline. And what we also had measured, which we don't show here, is the cytoskeleton was severely compromised. In addition to that, what we see is a larger population of the FGF treatments in the red dots. So we see the overall proliferation of that agonist having an effect in this data. And again, a very clear differentiation of the subpopulation. So what's unique about this is another method of analysis to differentiate subpopulations that you would probably never be able to get with a traditional flow cytometer. The next application that I'll overview is an extension of our IsoFlux product, which is used for isolating circulating tumor cells. So IsoFlux is a microfluidic design system that is used and designed to capture rare cells in whole blood samples. So the assay protocol was a whole blood assay, used two colors. In this case, we actually used DAPI to stain the nucleus, and cytokeratin, which is used as a cancer indicator. The samples were taken directly from the IsoFlux output, and the workflow is automated to scan the slides. So these were not used in a BioFlux plate. These are a simple slide that were used on a Zeiss microscope. The slides were scanned, the data reported with automatic report generation, and the overall analysis to count these CTCs from our IsoFlux samples went from days to hours. So the primary gate, which I show on this first slide, shows the average nuclear signal with the DAPI signal on the y-axis and the cytokeratin signal on the x-axis. So exactly what we would expect to see. Most of the cells that came from the IsoFlux samples would be normal DAPI signal, which is what you see in the upper left-hand quadrant. But what we would also want to see for these circulating tumor cells would be a high signal of cytokeratin indicator. So the upper right-hand corner shows a relatively small percentage of cells that would be initially viewed as potential CTC candidates. But the data analysis doesn't stop there. We also continue to look at morphological characteristics of those individual cells. And this is the real power of image cytometry and how imaging systems can be used to provide an insight into data analysis that a traditional flow cytometer could simply not provide. So in addition to that initial intensity gate, we also used qualifying gates to look at the cytokeratin signal intensity, as well as the shape and the ellipticity of the nuclei, as well as the overall size of the cell and the nuclei. So those are characteristics, again, that could never be used or looked at or viewed in a traditional flow cytometer. So what this allows us to do is to avoid false positives and negatives that would be generated with flow cytometry data. So for some of our customers who come to us for services with IsoFlux, we consider this analysis to be absolutely critical in determining patient samples and counts of the samples that we get from our customers and our collaborators. So as an output from these individual gates, we have automated simple summaries, which we see on the right-hand side. As you can see, the total sample has roughly 2,200 cells and individual events, and the number of cells that are in each one of the other subsequent gates. And at the very bottom is the final CTC count of 16 circulating tumor cells in that sample. As part of the other extension of that final report, this report provides us with the individual cell that's been identified, the gates that were used to qualify and count that individual event as a CTC, some statistical data about that individual event, such as the area in both signals, the intensity, and these are just a few of the data sets that can be provided. And then composite images to provide a cell gallery so that the user can look at the final report and determine ultimately what does this individual cell look like. And the last piece, before I hand the presentation over to Sean Burke of Novo Software, is a visual verification of those individually gated cells. So this is an extremely powerful feature that is part of the FCS Express. When the images, as well as the data that come from the imaging software, are handed off to FCS Express, both the data and the images can be used interactively. So ultimately what you have is a mosaic of all of the images that are scanned in both channels, both the DAPI for the nucleus and the cytokeratin cancer signal. They can be put together and assembled in a mosaic that can then be viewed and magnified to verify the individual tumor cells that were counted in the final CTC gate. Again, a very powerful feature that a traditional flow cytometer could simply not provide. Okay, with that, what I'd like to do is hand over the presentation to Sean Burke from Novo Software, and Sean will go through some information regarding the FCS Express software and some of the individual features and overall capability of FCS Express.
Hi, everybody. Thanks, Tom. All right. So, as Tom has mentioned, Novo Software has partnered with Fluxion Biosciences in the last few months to start providing this unique way of analyzing image analysis data just like flow cytometry data. And what I'd like to do today is take you through a little bit about FCS Express for image cytometry, so you understand a little bit more about how FCS Express works, and what some of that data looks like in real time, the data that Tom showed with the circulating tumor cells assay in FCS Express. But let me first start by telling you a little bit about Novo Software and FCS Express. FCS Express was developed as a powerful flow cytometry data analysis and presentation software tool. And in the last 2 years, we're proud to release our compatibility with image cytometry data. The reason that we did this is because the main principles of flow cytometry and image cytometry overlap. Things like taking multi-channel fluorescence data and plotting them in scatter plots and histograms to look for things like statistically significant differences and changes. And what FCS Express can do is now take that image cytometry data a step further by leveraging all of the most powerful analytical tools that we've built into our flow cytometry software to analyze those multiparametric data sets. And in the end, FCS Express is going to be able to relate those cell-by-cell results back to the original images, back to the single-cell level for confirmation of those results. Now, I just wanted to give you a brief company history just so you can see where we've come from. FCS Express was developed about 15 years ago, and it's been primarily a flow cytometry research package until 2 years ago in 2011. That's when we released our FCS Express Image Cytometry package, which is going to allow you to, again, take advantage of all the flow cytometry tools that are available within FCS Express. And I'd like to point out that we're actually in FCS Express right now. You may notice that this looks and feels kind of like a PowerPoint presentation or a Microsoft Word document, and that's exactly by design. That's because everybody knows how to use Microsoft Office. So if you've ever used PowerPoint, you should be able to come into FCS Express and analyze your image cytometry data from any of Fluxion's or from Fluxion at any time. And the way that FCS Express works is very much like PowerPoint itself. You notice here we have a ribbon and tab system, and in this ribbon and tab system there's many options for features in the software. But what we're going to cover today briefly is how to insert a plot and what that data looks like, and then we'll take a look at some of the CTC data that Tom had mentioned. So in PowerPoint, what you would do is insert a text box or a picture or a shape, but in FCS Express, we're going to insert data like a dot plot or a multi-cycle DNA cell cycle plot like you saw in the previous slides, or even a histogram. And to do that, we click on a plot, we draw a box for that plot. You notice that a plot is drawn, and we can just click and hold on any of the axes here to choose any of the different measured parameters, whether they be intensity parameters, whether they be morphometric data. Whatever is put out in your particular experiment as a parameter, we can choose that. So if I choose average intensity of the DAPI versus average intensity of the FTSE, you see that we have a two-dimensional plot plotting that data. And in this particular case, we probably want to change the scalings of this plot to a logarithmic view. It's going to make it look a little bit nicer, or it's going to allow us to work with the data a little bit more easily. So what we would do in this situation is right-click and format. And right-clicking and format in FCS Express is going to be one of your best friends, because what that's going to allow you to do is come into any plot in any situation and start making some changes to that plot, which means that you're not going to have to make any changes after the fact. All of these can be applied in FCS Express. And what I'm doing here is changing the x and y axes to logarithmic. And you can see now that that data is logarithmic. It's much easier to view. And what you may be used to in the imaging world is finding populations of cells based on a threshold. But in the flow cytometry world, we have something very similar, which is called a gate. And a gate is essentially a two-dimensional threshold. So if I come to my gating tab and choose rectangle, what I can do is draw a rectangle gate on this plot. You notice I'll be prompted for a color and a name, and I'll click OK. And what this gate is going to do is set up a threshold on the x and the y axes for the cells we'd like to include in this particular part of our analysis. What also is going to happen in FCS Express is we're going to be automatically presented with a certain statistic. In this case, we're looking at percent of cells that fall within that gate. But these statistics are live updating, and if I were to double click on this, I can change to a number of our default statistics that are within the software. And as I move this gate around, you'll notice that statistic updates. And what this is going to allow us to do is start diving deeper into our data and finding or looking for different populations that fall within many different gates, or populations or phenotypes of interest. So for instance, if I'd like to look at the cells that fall within gate 1 in another plot, all I have to do is drag that gate out, drop it on the layout, and you notice that now we're only looking at the cells that fall within gate 1 on this particular plot. And that's called applying a gate in FCS Express. And if we'd like to change to another parameter, to maybe look at the average intensity of the TRITC cells, we can now see the intensity values for those TRITC positive cells, and we know that they're DAPI positive and FTSE negative already. So we're starting to get some multiparametric phenotyping information out of this rather quickly. And at any time, if you want to get some statistics out of this, you can just right-click and choose Gate Statistics, and you'll be presented with a default statistics window to quickly get some information about how many cells fall within these gates. So that's a very basic overview of some of the things that we can do in FCS Express. What I'm going to do is delete some of this information and actually look at a CTC assay that Tom had mentioned. And we'll look at this in real time in FCS Express. So what Tom had shown is that the primary gating for this data was on this average intensity up here, which is in this upper right quadrant. And what we did in FCS Express, which is a nice thing you can do with any of your data, is you can create a histogram for that data as well. And in this particular histogram, we're looking at the average intensity of all the cells that fall in this average intensity gate. And we're also looking at some of the max CK signal over here in green. And what you can notice is that as we move this quadrant around, or as we move this gate around, we're actually going to update this histogram in real time to reflect our changes as we review the data. So we'll be able to quickly and easily see what changes to gates actually do to some of our other parameters in the data sets. Tom had also mentioned that there's lots of qualifying gates in this particular assay. You can see we had the max CK signal gate, the area gate, and the shape gate here. In this slide, what I want you to notice is that we don't just have to use these default gate statistics windows that pop up. We can make this analysis look and feel any way you like by displaying statistics in what we call, or what are known as text boxes in FCS Express. And if you were to make any changes to any of these gates, you can notice how these statistics are going to update for you in real time. So again, as you mine through your data, you'll have live updating statistics. We also mentioned that we can relate all of the images back, or all of the plots back to the actual images. And what we have here is a mosaic image on the top with the black background from the assay, and all the cells or all the events that were segmented are highlighted with the different colors from our gates here that we've created. And in the bottom, what we've done is we've removed the black background to get a little bit more contrast so we can see what's going on. And what this means is we can actually apply one of these gates, like the area gate, to this plot. And what this is doing is just showing us all of the cells that fall within this area gate. We could do that again with the shape gate here. And you'll notice as we apply that gate, as we move the gate around, all of our pictures and all of our plots are going to be updating in real time. So we're starting to be able to get spatial information directly from the plots itself. And what we can do in the end is actually take those large mosaic images and finally break them down to a cell-by-cell level. And you can see that we've done that here with each of the individual cells in what we call a cell gallery or a data grid. We're also enumerating those cells over here in this table. And a very nice thing about these single cell cutouts is that any time I can come in here and edit this gate. And if I wanted to visually confirm, I know that maybe this is half of a cell and this one is half of a cell, and there's bits and pieces here that I want to remove, I can quickly and easily remove these cells from our analysis, and you'll notice that not only do they remove them from our data grid, but our counts in the end are also being changed to reflect all those gating changes. And now these gates can also be applied directly to this data grid as well to view events in different gated populations that you've created. All of these small boxes here are indicating which gates these cells are coming from. So there's lots of powerful tools, again, to move from a population-based analysis down to a single cell analysis. And what I'd like to summarize with today is that FCS Express Image is going to leverage all of these powerful tools from Flow Cytometry for your image cytometry data, all with a Microsoft PowerPoint-like feel, and you can easily move from the population level down to the single cell level at a click of a button. So if you'd like to try out FCS Express, you can get a free demo from our website at www.denovosoftware.com. From our download section here, you notice you can get this free demo for 30 days. And if you have any questions for the team after the webinar, feel free to contact us at support@denovosoftware.com, and we'd be happy to answer any of the questions that you have. So I'd like to hand this back to Tom. I think we've given a quick overview of what FCS Express has to offer. So I'm going to hand this back to you, Tom.
Thanks for that overview, Sean. That was fantastic. Hopefully everyone has a good appreciation for the power that that capability provides. Very unique, very strong, certainly very tremendous data that can be analyzed from a number of different perspectives. Just a few more slides to close things out, and then we'll open it up for questions from the group. Last thing I wanted to just quickly review is the system can include a number of different product configurations. The system can come configured with a fully automated microscopy system. What we do resell, we are an authorized Zeiss reseller, so we can sell Zeiss axioscope microscopes with our BioFlux 1000 systems, and in addition of the CytoFlux software. In addition to that, we also have the BioFlux 200, which is a manual microscopy system, and the CytoFlux software can be simply added to that. And then we also have upgrades for our existing BioFlux customers. And then in addition to that, we can also provide upgrades to Metamorph users. So Metamorph is an image analysis software package from a company called Molecular Devices, and we can simply add a software upgrade to their capabilities and allow a lot of this functionality from existing imaging data sets. So quite a few different options and ranges there. Comparing quick summary of the feature set, by taking advantage of your existing inverted microscope, CytoFlux can provide you with the highest image quality in a cell analysis tool. We can minimize the dead volumes, allow you to look at adherent and suspension cells, the ability to look at live cell kinetic data, as well as the ability to look at 3D data analysis. In the next month or two, we'll have another webinar that will show the analysis of different 3D data stacks from confocal images. And you see the complementary functionality and features to a traditional flow cytometer. The system is really quite unique and very different from what you would see in a traditional flow cytometer. So hopefully what I've shown everyone today is how the CytoFlux system can turn your inverted microscope into a world-class imaging cytometer with uncompromised image quality, the ability to look at various different cell types, minimizing your dead volumes, and in the end, the ability to recover your samples. So with that, thank you for attending, and I would like to open up the floor to any questions that someone may have. While we're here, I'll bring up just our CytoFlux page. One quick application that I did not review today was the ability to look at different suspension cells. So what you can do is go to our website where we have some data and good examples of Jurkat cells that were imaged using our BioFlux plate and our stop flow protocols. The Jurkats were stained and loaded into a BioFlux plate, and the cells were flowed through the microchannel, stopped, imaged, and then so forth and so on. Ultimately, roughly between 40 and 100 different stop flow sequences would generate thousands to, in some examples, tens of thousands of cells. Looks like we've got our first question. First question is price ranges. So I had some price ranges on a previous slide. Let's see if I can go back to that. All right, here's a second question. Second question came in and asked, how was the data from the HUVEX imported and used to generate the cell cycle data? So that data was actually, I think it was very convenient, and it was literally, once the data is transported and exported into FCS Express, the process was as simple as checking off a multi-cycle checkbox and then selecting which parameter was used to generate the histogram, which we showed. And in that case, I believe we used the average nuclear intensity, and it was as simple as that. The only other addition and change to that plot was I normalized the y-axis to the counts, and the data was populated immediately into that graph. So that was one of those data examples and application examples where the data was populated almost automatically, and there was very little change to the graph that was required.